Instead, I chose something different. I chose the impossible. I chose rapture.
You could spend hours exploring the world of Rapture, whether it be getting lost in the detailed Art Deco architecture or collecting all the countless audio diaries. It is undeniable that the world of Rapture is rich in detail and backstory. But I think one of the most fascinating aspects of the game is the emphasis it places on science and technology, and in particular, genetic enhancement. In the opening moments of the game, we are very early on introduced to the concept of genetic modification.
Steady now. Your genetic code is being rewritten. Just hold on and everything will be fine.
Well, there are two major components in the game which allow for dramatic genetic modification: Adam and Eve. Eve is a fluorescent blue liquid found scattered through the world of Rapture in the form of injectable hypodermic syringes. Its presence is more of a game mechanic than contributing to the backstory of the game, and hence I won't be discussing it in depth in this video. You can think of Eve being similar to mana in most role-playing games, and its purpose is largely similar, primarily to stop you from spamming your plasmids. Adam, on the other hand, is a processed red substance extracted from sea slugs, which promotes the formation of potent stem cells. As Yu Su Chong described it, Adam is a canvas for genetic modification. The exact mechanism in which Adam operates is at least partly elucidated through the audio recordings of Bridget Tenenbaum.
Adam acts like a benign cancer, destroying native cells and replacing them with unstable stem versions. While this very instability is what gives it its amazing properties, it is also what causes the cosmetic and mental damage.
But what exactly is Adam? It's important to note that Bioshock was released in the year 2007, just a year after Shinya Yamanaka's lab at the University of Kyoto was able to convert fully differentiated adult cells to pluripotent undifferentiated stem cells. He did this by isolating adult cells and adding certain factors. These chemicals are found at relatively high concentrations in the developing embryo, and trick the cell into reverting to a more primitive state. These findings were groundbreaking for the field of stem cell research, which had previously relied on embryonic stem cells, a subject viewed as controversial and unethical by some. As a result, Yamanaka was jointly awarded the Nobel Prize for Medicine in 2012 for his work. There is no doubt in my mind that these findings at least partially influenced the development of Adam and the mechanism by which it operates. The seemingly endless possibilities of Adam sparked interest in scientific and business communities alike. The mass demand for the substance necessitated a new means of production. You couldn't simply get the required amount of Adam from sea slugs anymore. Tenenbaum realized that if these slugs were implanted in the bellies of prepubescent little girls, she could exponentially increase her supply of Adam, leading to the creation of one of the game's most iconic characters, the Little Sisters. So we know about the extraordinary capabilities of Adam, and how we can get it. But how exactly can we use this to enhance our everyday lives? Well, that's where plasmids and gene tonics come into the equation. Plasmids are the most ostensible display of the capabilities of Adam throughout the game. Traditionally, plasmids are small circular loops of DNA found floating inside most bacterial cells. These regions of DNA normally encode virulence factors such as antibiotic resistance. But let's say we wanted to make our own plasmids for human transfection. First, you'd either have to find an organism with the desired gene in nature, or be able to synthesize the gene from scratch. You'd be hard pressed to find anything remotely close to matching the destructive power of plasmids showing in the game, occurring in nature. But, to be fair, if the game limited plasmids to only those found in nature, it would certainly be a lot less enjoyable to play. However, you'd still have a problem of getting the genes into human cells. Plasmids are fine for inserting genes into bacteria, but humans don't have them floating around in our cells, and even if we did, they're far too small to house large and complex genes. I don't think the plasmids in the game actually represent plasmids in real life, but rather they just lend their name to them. I think the most likely explanation is that the developers just glossed over some of the facts and took some creative liberty in this regard. It is just a game after all. A more accurate comparison would be something like viral vectors, which are already used in gene therapy. Viral vectors work on a relatively simple premise. First, we take a deactivated virus and insert the gene of interest. We then reintroduce the transvected virus into the host. When the virus replicates, the inserted sequence of DNA integrates itself into the host. Overall, I think the developers of the game should be commended for their efforts. Bioshock is a game which isn't entirely focused on science at its core, but yet is resoundingly rich in detail and for the most part holds true to the breakthroughs in scientific research, unlike most games today.